All right. Hello, everyone. My name is Natyelli Lopez, and today I will be discussing and presenting my long talk on microbial extremophiles as a model organism to discovering life beyond Earth. Okay, so for my outline, today I'm going to be talking about what exactly extremophiles are. I'm going to be giving an in-depth explanation of them. Then I'm going to move on to why studying extremophiles matters, specifically on furthering space exploration, finding life beyond Earth, and also protecting life within Earth. And then lastly, I'm going to give an introduction to an academic journal that deals with extremophiles and using extremophiles as a model organism to discovering life beyond Earth, in particular, the planet Mars. Okay, so to begin my presentation, what exactly are extremophiles? So all throughout Earth, we can find areas of high heat, extreme cold, high salinity, high radiation, and high acidity, etc. All which seem kind of inhospitable for life, but there are organisms that not only survive in these kinds of environments, but thrive in them, and these are known as extremophiles. So the word extremophiles derives from Latin origins, and it means extreme loving. So it's very fitting for these organisms to be called extremophiles, but it is also a pretty vague term because it varies from all different domains of life, like bacteria, archaea, and eukaryotes. They also differ in different kinds of environments and areas that which can be found, in which they can be found throughout Earth. One of the most extreme forms of extremophiles are known as tardigrades. So most extremophiles are known to withstand just one kind of extreme environment, but tardigrades are known as the most extreme, also known as water bears, because they can withstand different kinds, up to multiple kinds of harsh environments. And so they're going to be really important, especially when it comes to furthering space exploration. And I'm going to be talking about that more in a further slide. All right, so moving on, what kinds of and the types of extremophiles that can be found throughout Earth. So there are many different kinds. Some examples include pisophiles, which can withstand high atmospheric pressure. Many of them can be found at the bottom of the ocean near trenches because the pressure is so high. And the reason they can withstand such high pressure is due to the fact that the amino acids within them have taken up less space within their cells. And so it prevents the proteins to fold in on themselves and essentially crushing them to death. And so since they have these proteins that are can avoid having them being folded in, they can survive and reproduce in these kinds of harsh conditions. Then we also have halophiles, which can survive in areas of high salt concentration. These are studied in places, especially in the US, like the Great Salt Lake, the coastal regions near the ocean, which have high salt concentrations in the water. These kinds of organisms can survive high salt concentrations due to the fact that they have organic compounds called osmoprotectants. And what these osmoprotectants do is prevent these organisms from having the salt come inside of their cells and essentially drying it up and shriveling it. So it prevents them from being shriveled up to death. Then we also have psychrophiles, which live in extreme cold. And I've pictured them here. They're kind of like these ant-like structure or there's ant-like organism. And they can withstand extreme cold due to the fact that they have proteins that act in the way that antifreeze does. And so say you were to put an organism like a human into the types of extreme cold and temperature that these organisms live in, you could potentially put that organism at risk of crystallizing the cells, and which would eventually lead to death in that organism. And so these organisms have developed these antifreeze-like proteins, which prevent them from being killed by this extreme cold. And then lastly, I have thermophiles, which are going to be really important, especially when it comes to the academic journal that I'm going to be talking to you guys in a later slide. These organisms live in extreme heat, so they can be found in areas with volcanoes, in areas that have hot springs, and can be found in the desert. And the reason that these organisms can survive such high heat is due to the fact that their proteins are folded and compacted so tightly, it creates kind of an elastic effect. And this elastic effect prevents it from essentially folding open and killing the organism. And so that's the reason these organisms can survive in such extreme temperatures. And these are only to name a few. There are different kinds of extremophiles. There are many kinds of extremophiles that would live within different kinds of extreme or harsh conditions, but these are mainly the ones used in science, especially the biological sciences. And many of them are used within the study that I'm going to present to you today, like the thermophiles that I mentioned before. All right, so moving on, why does studying extremophiles matter? So down here below, I pictured a tardigrade, or known as a water bear, like I mentioned before, because of their ability to withstand multiple kinds of environments. They were hand-selected by NASA scientists to be sent out into space, and they were going to study the genetic material or identify these kinds of mechanisms that the tardigrades use in order to survive and withstand these harsh and high stress conditions. And so they did this in hopes of finding out information which they could use to send astronauts out into space and reducing the amount of stress, especially when it comes to long duration space travel. And so extremophiles are really important because they not only help us find life beyond Earth, but it protects the life within it and those that want to travel outside of it as well. All right, so secondly, as to why studying extremophiles matters. So due to the fact that I mentioned before that they are able to withstand different kinds of harsh environments, they could be used to replace catalysts and speed up reactions. Many of the catalysts that we know have certain environmental limitations, which prevent us from using them in certain processes. And so what scientists have been able to do is utilize the genetic material of these extremophiles and place them with things such as enzymes, proteins, and other organic compounds that could be used to replace these catalysts that were previously unavailable to them. Also, doing this, it helped them reduce the amount of spending that comes with creating this research. Usually, research with catalysts are extremely time consuming and very expensive. And so they were able to utilize this and speed up the process and make it make it inexpensive. And so thanks to this DNA of the extremophiles, processes like PCR, which can be used to amplify the genetic material of a certain organism so that scientists can study, was able to be made. Then there was also the creation of biofuel. That's also thanks to extremophiles. And then we can also say that thanks to extremophiles, we have been able to clean up radioactive waste through biomediation. So that's one of many reasons why studying extremophiles is extremely important. All right, so now that I've given a little bit more context as far as what extremophiles are, I'm going to be presenting a research article or journal that was led by Susanna O.L. Dorito, and it's called A Wide Variety of Putative Extremophiles and Large Beta Diversity at Mars Desert Research Station. And essentially what they wanted to do with this experiment was collect soil samples, from an area in Utah called the Mars Desert Research Station, or Mars Desert Research Site. And from this soil that they were going to collect, they were going to determine what kind of life they found within that soil and determine if it was plausible to be able to live on Mars. And so the reason that they picked this specific location was due to the fact that it was the most similar to Mars-like conditions on Earth. And so obviously there's things such as oxygen that vary and pressure and other things that come with being on a different planet, but it was considered to be the most similar to that of Mars conditions. And so they were going to determine what kind of life they would find here and see if it was able to live also on Mars. Right, so for a little bit more context to this experiment, like I said before, it was going to be done at the Mars Desert Research Station or site in Utah. So here I've provided a map of the states, and here's Utah. And this little gray region here is where the Mars Desert Research Station was set. And here is this region here that they were going to collect the soil samples from. And these numbers are the specific locations where they were going to collect them from. And the reason that some of them are scattered and spaced out is due to the fact that they were going to collect soil from different sections in the same region, because it was still similar as far as atmosphere, temperature and atmospheric pressure to Mars, but it was different kinds of soil samples, like different material. And they were going to determine what kind of life and if that varied as far as finding biological material within that those samples. All right, so for the hypothesis in this study was very clear. They mentioned that one of the main objectives was to find life within this region, determine what kind of life it was, and see if it was able to survive in Mars-like conditions, and see if this kind of life that they find within this desert-like region was similar to that of other organisms that were found in other research studies that involved extremophiles in desert conditions. And they also wanted to determine how this could relate to Mars-like physical and chemical parameters. Okay, so for the methods and on-site experiment. So the methodology was a bit tedious and complicated. So what I did here was create a flow chart to kind of simplify the processes that they were, or the methods that they were going to take in order to complete this experiment. So I'm going to be going over that with you. The first thing that they did, like I mentioned before, was collect these soil samples from the Mars Desert Research Station. And they did this around January to March of 2009. And they were going to collect soil samples from 4 different regions of this Utah site, due to the fact that there was different kinds of soils, and they wanted to see if the type of life within those soils varied. Then once they collected those samples, they were going to extract the DNA, and they used 2 DNA kits to extract this DNA and from the soil samples that they collected. And I believe they collected 14 soil samples, and so they used 2 DNA kits for each. Once they received the DNA from the, or what they could, once they received what they could from the DNA kits, they moved on to using 16 16S RNA gene fragments for bacteria and archaea, and then 18S RNA gene fragments for eukarya, and they amplified this by PCR. And so we know that PCR is used to amplify the genetic material of a specific organism so that scientists can study the genetic material or the genetic makeup of certain organisms. And so once they received this PCR product, they profiled them by denaturing gradient gel electrophoresis. And so many of the genetics students might understand this process. And so once they placed the biological material, All right, so um, they once they received the material, once they placed the genetic material inside of the inside of the gradient gels, which were made of, I believe, 8% polyacrylamide. The denaturing gradient for bacteria was 30 to 50%. For archaea, it was 30 to 70%. And for eukarya, it was 20 to 35%. Then after they got that, they stained each gel for each individual soil sample that they got with ethidium bromide. And the ethidium bromide was essentially just, you use that in the denaturing gradient gel to optimize the visuals that you get from placing the gels under a UV transilluminator and photographing them. And once they photographed each individual gel for each individual soil sample, they used a DGGE analysis, and they were converted and analyzed and normalized with a gel compar2. Once they obtained this, they calculated the similarity values using Pearson's correlation and dissimilarity coefficients, and they were visualized with arithmetic means cluster analysis. So once they completed this, they performed the same processes 1 through 6 over again to make sure that their data and their information was clear. And so once they finalized this and received the sequences from the biological material that they found, they placed it under the gene bank and used BLAST to determine what kind of biological life they found within these soil samples. All right. So for results and analysis. So it was clear that from the information and from the soil samples that they got, all life from all 3 domains were found, so from archaea, bacteria, and eukarya, but it wasn't found in all of the soil samples that they collected. So down here, I provided a chart that they gave in the article. They collected 14 soil samples from different locations. And from those different locations, they determined whether the biological material that they found was either bacteria, archaea, or eukarya. These plus signs down here meant that they did find biological material from the certain domain of life, and the negatives or the minus signs means that they did not find it. And so from all of this information, we can see that some of the soil samples were they didn't have biological material, but the most present could be seen down here with the total positive. So we can see that bacteria was the most present, with archaea coming in second. But it did mention that even though archaea did come in second, it was the least diverse of the 3. And then eukarya came in last, with being the least present within the soil samples that they found. Also, using the DGGE analysis, they profiled and saw that the highest was alpha diversity rather than beta diversity. All right, so just for a little bit more context from their results, here is the picture that I provided earlier of the region that they were going to perform this experiment. So these numbers here were where they collected the soil samples. And using the information that they received from the gene bank and BLAST, they profiled the biological material to be certain kinds of bacteria. And here's the different kinds of bacteria that they could have found, and the pie charts of the amounts of this kind of biological material that they could find within these certain regions. And so just for a little bit of context, I thought this was really cool. Okay, so in conclusion, and my take-home messages. All right, so what I basically wanted to state in this presentation was what exactly an extremophile is and what's its function and why it's important to study extremophiles. And using that information of what an extremophile is, explain to you this this research that was done to find life outside of Earth. And so from this experiment, it's important to note that all domains of life were detected in this Mars Desert Research Site. And so it could be determined that even though there's only 3 options for life, it could be a possibility to find all 3 on places like planet Mars. Also from this research, it's important to know that even though there's soils that differ from different regions, there's still always biological material found within those different regions, and it can vary even though they're similar. They're similar in their kind of like geographical stance. And lastly, using the methods that they use, like the PCR, the DNA extraction kits, and the denaturing gel, gradient gel, we can use those kinds of methods to determine if we can find life on planet Mars as well. And so I thought that it was important to mention why finding life outside of Earth would be relevant to us, because as we can see, there's a lot of problems with like food shortages and space among humans. And so if we were able to find life and cultivate things such as food on a different planet, we could definitely use that to our advantage to keeping humanity alive. So thank you so much for listening to my presentation. These are